From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 27, recorded on January 12th, 2018. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today, right here in New York City, Nels Elde.
Hey, Vincent. Great to be here in vivo.
Yeah, it's good to have you.
Thank you so much.
Evo, Devo, and Vivo. We could go on and on with that.
I think we probably will.
And what brings you to New York City here?
Yeah, so I got in Wednesday afternoon and was hosted at NYU in the Department of Microbiology. Victor Torres, who is a spectacular microbiologist, was my host. And so yesterday I started early and went late into the early morning hours. You might detect that in my voice, which might be slightly rough around the edges. But what a fun day of science and giving a seminar at NYU in microbiology and just connecting with both old friends and hopefully making some new ones.
Yeah, it's always great to visit, right?
So fun.
Yeah.
Yep.
And I'm glad you decided to come up here and do a couple of podcasts.
Well, you know, I couldn't resist.
In the same city, right?
Yeah, exactly. Back-to-back podcasting for us today.
Yeah, so we're going to do a TWiV later on.
Mm-hmm. I'm excited for that.
I picked one paper and kind of hoping it's up your alley for that.
Fantastic.
And then the other one is just really cool, so that should be fun.
So what's the latest from here at TWiV headquarters?
Well, let's see. What's going on at TWiV headquarters? We're just podcasting away, you know.
Mm-hmm.
We just want to get bigger and bigger, and I'm trying to figure out how to do that. I'm trying to figure out ways to sustain this endeavor.
Yeah.
And, you know, up to this point, what do we have? TWiV, TWIP, TWIM, TWiEVO, Immune Urban Agriculture. We have 6 podcasts. It's almost about as much as I can do.
Yeah.
But I want to get some other people involved and help out and so forth. I'm trying to figure out how to do that.
Well, I couldn't help but see a couple of days ago that the Washington Post
that was really nice, yeah. They had an article about This Week in Virology, and, you know, they said viruses are important parts of our lives, and people need to know more, and this podcast goes into it, and it's a must-listen, they said. So it was really nice. You know, that just came out of nowhere. Of course, a lot of people see the post, right? So I got lots of emails.
Good.
I just wish we could get more similar coverage and for all of our work, right?
Sure.
Because it's not that I want to make money. I just want to spread the word.
You and me both. And I think that idea of if you build it, they will come. Hopefully that will continue to happen.
That's been my philosophy. I've bbeen very patient. Mm-hmm. I think if you make a good product, people are going to listen, right?
Agree.
And I know science is hard, and we're not going to have as many people listening as listen to other kinds of podcasts. I don't know if you've heard of this podcast called Pod Save America. Mm-hmm. It's a political podcast, right? Mm-hmm. They get 1 to 2 million downloads per episode. You know, I'm happy on TWiV to get that in a year with 52 episodes.
I hear you. TWiEVO is even a little more modest, a few logs down.
Well, you know, there are thousands of people listening.
Yeah.
And they love it. So we do it for them. And that's fine. You know, if 10 people were listening, I don't think we would do it. But thousands is fine. But I would like to reach hundreds of thousands of people. And I think you have to have a different kind of product. You know, you have to be Howard Stern, shock jock, to reach hundreds of thousands of people. And I guess you could do that for science, but you and I can't do that. So
agree. Although the science itself is increasingly shocking, I think, especially in the evolutionary arena, where we see all of this incredible work coming from all corners and uncovering these incredible evolutionary mechanisms in ways that you might not expect. And for me, that's shocking and exciting.
No, it's totally cool. The thing is that it's hard, right?
Sure.
When we talk, it gets complicated. And even the Washington Post author, she said, it gets complicated and you might not understand it all. So I'm wondering at some point if I, together with some other people, make something that is totally much more accessible, right? We really simplify things. Maybe that has a broader appeal.
Yeah, it's a really interesting point, and one that I certainly struggle with. Not only podcasting is a sort of emerging rookie here, but also in general audiences, specific audiences, even audiences in different departments.
Yeah,
So
sure.
if you're talking to a genetics department or a microbiology department, different backgrounds, different crowds. And so finding that balance between the details and the big points, that's a journey.
The thing is, we have a lot of people listening to TWiEVO who are in the field or related fields. They're scientists, right? You can tell by the emails. And if you change it to be very simple, they won't like it anymore. But then maybe other people would move in. So that's why I think you need a completely different product.
Yeah.
I like going deep into science. I like taking as much time as we need. And there are people who want to listen to that, but not a lot of people are going to listen to 2 hours, right?
Correct.
So I think if you had a shorter product that went over things in much simpler ways and explained why it's important, for example, that would be. And so I'd love to do something like that one day with someone out there.
Yeah.
But I do have a lab for now. I mean, the key is at some point I'm going to stop the lab and do this full time, and that may be this year or it may be next year. I just don't know yet.
Yeah.
And when I do, I don't know where I'll go to do this. I don't know if I can stay here. So I'm trying to figure these things out. I'm trying to see if I could raise money to, make a permanent home for podcasting, maybe here or somewhere else. There, other places are interested in me going there and doing this.
Yeah, fantastic.
If Columbia isn't, I will go somewhere else. I have no problem. Yeah. I'd love to go somewhere and, teach, write, podcast, communicate for the next 5 years or so, and establish something that goes on beyond me. I'd really like to do that.
Yeah. This all sounds really exciting to me, and I can't wait to see what happens. Stay tuned. And in the meantime, if you're out there listening, please think about helping support the efforts on all This Week in Science, This Week in Evolution, Virology, Microbiology, on and on. Please help us out.
Microbe.tv/contribute.
Yeah.
And by the way, we're giving a book away today, Nels.
Oh, interesting.
Forgot to do this last time. I wanted to do it over the holiday. Our last episode was with Vinnie Lynch, right?
Correct. Yep.
And so Michael Cordingley wrote a book called Viruses, Agents of Evolutionary Intervention.
Oh, wow. I haven't seen that one.
Invention, not
Invention.
intervention. Mm-hmm. And he listens to podcasts because he wrote me a couple of months ago. He said, I have a couple of extra books I'd like to give them to you.
Oh, fantastic.
And he gave us one for TWiV and one for TWiEVO. And he signed them. This happens to be the TWiV signing, but he wrote, "Dear TWiEVO listener," and and he talked about the podcast. Regards, Michael Cordingley. So,
oh, wow.
At the end of today, we'll give this away. We'll call for a contest of some sort.
Great idea.
Usually on TWiV, we just say write in and we'll pick a number at random. And a couple of weeks ago, Islam Hussein, our friend of the show who does Viral Vlog, he said, you should do something different. Why don't you think about how about a crossword puzzle? Have people solve it. So for TWiV a couple of weeks ago, I made up a crossword puzzle, or I took one from Ann Palmenberg.
Oh, good idea.
And no one's
filled it out yet?
No. Actually, the only person who submitted it just today, Amy, my person in the lab, she finished it. And so no one else has, so you give people a little work, they don't answer the contest.
Right.
So
maybe the drawing a number out of the hat is the way to go here.
But, I was thinking if you could get like 2 people to subscribe to TWiEVO, you can enter the I don't know how to do that, though. Right?
Yeah, we'll have to think.
I'm not sure. But,
let's think about that.
But later in the show, we'll announce this contest and how you can enter.
Fantastic.
But you picked a cool paper for us today.
Yeah, well, so, coming from wintry Salt Lake, it's actually been quite warm, unseasonably warm, and unfortunately a little bit dry. So we're behind schedule.
Now here it's been bloody cold. Right. Has it been really, really cold in Salt Lake as well?
No, so when that polar vortex sort of descended down in the east, the west, there's been a high pressure ridge that just sort of set in and just sort of hangs out, which is okay. It's warm, but it's also the weather just gets really boring. So, and by that I mean there's not enough snow for the skiers and the snowboarders and the outdoor action. So anyway, with that in mind, I was happy actually to come into this potentially cold scenario, the polar vortex. I missed it. It looks like it's almost tropical today out there.
Today, it's crazy. It's warmed up. The snow is melting. It's generating fog everywhere.
Yeah.
Driving in today, there were parts that was like soup. You couldn't see anything.
Is that right? Yeah.
And it's, let's see what the temperature is here.
So we're sitting here at headquarters looking out over the Hudson River, and there's just steam coming off of the river. It's really striking.
It's 14 Celsius.
Okay, yeah, quite warm.
Now, according to this forecast, Sunday's supposed to go back to freezing.
Okay.
And then we're supposed to have snow next week again. So I don't know if that's going to work.
So
have you done any skiing this year?
I haven't, because it's been so dry. It's actually, the fever is broken. So we got a couple of nice storms in the last week. And so I think things are improving nicely. But anyway, getting to this polar vortex, that was part of the motivation for today's paper on the evolution of cold sensitivity, or diminished cold sensitivity, in some hibernating critters. I thought this might be a nice token of warmth to bring to the polar vortex. And maybe
Totally. This is a cool paper. I love it.
Yeah, really interesting. So this is a paper from, I hope I don't butcher the name here, but Elena Gracheva, who is at Yale University in the departments of cellular and molecular physiology and Department of Neuroscience, or program in cellular neuroscience, on molecular prerequisites for diminished cold sensitivity in ground squirrels and hamsters.
Now remember the ground squirrel. This is the 13-lined ground squirrel, which was the subject of Hopi Hoekstra's paper, right, when she came on TWiEVO.
Exactly right. And in fact, that was sort of how I got connected to this paper, was a tweet from Hopi holding this up. And so
she liked this paper.
I think so. Yep.
I think it's just great that you can, as you'll see in this paper and also with the Hopis, you can do deep molecular biology and understand how things are working with different animals where we just used to catalog them, right?
Yeah, agree. And so that's right. And so here, I think a really interesting example of connecting some of the evolutionary biology, in this case, or independent or convergent evolution, where in two very different species, as you pointed out, Vincent, the 13-lined ground squirrel, but then also the Syrian hamster, very different, but have some, some interesting parallels in the evolution of their physiology to deal with cold.
Cold, right.
Yep. And then, as you're saying, excuse me again, connecting that evolutionary path all the way through to some of the neurophysiology, or at least some measures along these lines.
As you'll see, this paper is amazing, really.
Agree. Yep.
So let's talk a little bit about cold sensitivity, right? Because that's not our field, and we haven't ever talked about it.
Yeah, we haven't, and I'm glad we are getting to it, polar vortex notwithstanding. And the reason is that cold sensitivity turns out to be, it's not just the mammals, it's not just the hamsters and ground squirrels that care about this, but think for a moment actually about fish in and around the polar caps, actually. And so in evolutionary biology, this idea of antifreeze proteins and their evolution, these sort of extreme environments where creatures will find themselves or have adapted over millions of years in some cases, is a sort of striking phenotype, in a sense, is how do you deal with life right at the threshold of freezing? How do you survive this?
Right.
You can also think about other adaptations to extreme environments, high elevation, that can also be quite cold. Sure. And so this has been, for more than a generation, captured the imagination and attention of evolutionary biologists. And so cold sensitivity, there's not only in terms of hibernating critters like the ones we'll consider on TWiEVO today, but in other cases, such as these Antarctic fish, and how do you keep pumping blood when you have ice crystals potentially forming? Yeah. The molecular evolution of antifreeze proteins.
Yeah, that's amazing.
So this is amazing. Really fun area of active research. Part
of the idea here is that cold sensitivity means when it gets too cold for you, you seek out warmer areas, right? And there are, obviously, that's a sensing issue. There have to be cold sensing properties in animals to be able to do that. And then going beyond that, hibernating animals have to even have a lower cold threshold, right? They have to be able to deal with cold temperatures when they hibernate and not wake up and say, I'm cold and go somewhere
That's
else.
right.
Basically.
Yeah. Not panic when the cold hits you in a polar vortex.
Yeah.
There's a good parallel there somehow, right?
I got to tell you, last weekend here, it had snowed on Thursday, I think. And then the temperatures were way below zero for a long time. And to go outside was just painful. I have to tell you, I would walk my dogs in my backyard, and we have a little one, and she would, walk on the snow and then immediately come to me and want to be picked up because her paws, her foot pads were hurting on the snow.
Yeah.
And it was so cold, right? So obviously that animal doesn't have good cold sensitivity. But it hurt me, and I wanted to go inside. and you'll see in the assay that we talk about in a minute, that's relevant. So they say here there are cold receptors that are involved in detecting temperatures, right? And these are basically receptors on some neuronal population. And they say cold receptors account for 15 to 20% of the total neurons in the dorsal root ganglia of mice and many other vertebrates. So that means it's really important to sense cold, right?
Yeah, we've devoted a lot of horsepower to sensing this under conditions that could be the difference between surviving or not, right?
I wonder what the percentage is in us, right? So we're derived from the primate line, which typically lives in warmer climates, right? Although there are some New World monkeys that live in colder areas.
Oh,
right?
yeah. Oh, yeah.
I'm thinking of the ones in Japan, right?
Yeah, the snow monkeys.
The monkeys, yeah. But the ones in Africa, they're warm. So I would guess, and I don't know the answer, that we don't have a lot of cold receptors in us. Not 20% anyway.
Yeah, good question. I don't know the answer, to be honest, but I think that's right. I mean, in some ways, the primates, and our species included, have one adaptation would be to, when you're sensing cold, is sort of strategize a whole different way of living.
Yeah,
And so,
yeah.
Although at the same time, of course, there are indigenous populations that have populated the world. And so,
right.
Again, you can imagine tackling some questions and taking some of these other mammalian model systems where there might be some clues or some hints about thinking about the molecular basis of some of the sensitivity to cold and how you deal with that from an evolutionary standpoint.
Now, a main player in this paper is a channel called TRPM8.
Correct.
A cold-activated, non-selective cation channel. So that I don't know a lot about this, but it sounds to me like cold will activate this channel and lets cations, positively charged ions, come in, and that somehow causes a response. If it's too cold, you're going to go somewhere else, right?
Yeah. And so I think, that molecular handle, right, where it's, excuse me, it's probably pretty, you're the easiest thing to consider,
right?
Is that sort of first step where you sense something.
Yes.
And so I think that's where this paper gets really nice traction, is by taking this at sort of the first step and then considering it. Yeah. But then as soon as you hook this up to a more complicated neuronal circuit and then start thinking about the signaling that then somehow reads out or gets transduced into a behavior, that gets a lot more complicated.
That's hard.
Yeah, that's right.
No, this is the easy part, really, although it's pretty tricky as it is.
Yeah.
So they say this TRPM8 mediates responses to cold below 26 Celsius. And it's an essential part of the thermosensory apparatus. And it's not the only part, okay? So this is just part of it. But it defines the range of tolerance for a species and therefore where it's going to live, right? And they say an extreme example is demonstrated by hibernators that can withstand prolonged exposure to cold and hypothermia. And we don't know what kind of adaptations have led to that beyond thermotolerance, right? Normally running around in the winter and so forth.
Yeah.
And that's one of the things they'd like to do here. So that's why in this paper, the two animals, the 13-lined ground squirrel and the Syrian hamsters, are mammalian hibernators, right?
Exactly. And so you can imagine if you're hibernating and all of your metabolism sort of goes into that state.
Yeah.
At the same time, if you're not going to find a Syrian hamster in New York City, but you might find other hibernators. If the polar vortex hits and you panic and lose that hibernating status, you can imagine some fitness consequences.
Sure. Yep. Absolutely. So let's talk a little bit about this assay, which is an essential. They have a a two-plate temperature preference test. And when I read that, I said, oh, this is something complicated. But it's not. It's two plates at different temperatures. And they just put the animals in a cage with these two plates, and they see how much time they spend on either plate, and they have different temperatures.
Mm-hmm.
So it's a preference test,
Yeah.
basically. So you can have a reference plate at 30 degrees Celsius, and that's pretty hot, right?
Yep.
Or they can have another plate, which they will range from 0 to 30 degrees Celsius, right? And what they do is they put animals in, and they said earlier studies have shown that mice prefer 30 degrees over cooler temperatures, and they avoid temperatures below 10 degrees.
Mm-hmm.
So you can imagine using this two-plate thing to figure that out. That's a fun assay.
Yeah, I agree.
And then squirrels and hamsters have a preference to 30 only when the test plate gets warmer.
Right.
Like if you raise the test plate to 5 and 10, then they like to go to the 30. But they prefer, like at 0 Celsius, they don't go to the 30-degree plate at all.
Yeah.
So they like the cold, basically.
Yeah, almost like flipping a switch. Whereas the
That's
mouse species, which doesn't hibernate, has a very different phenotype.
Right.
And then again, if you think about this from an evolutionary standpoint, the ground squirrel and Syrian hamster are actually very distantly related.
Right.
And so then this gives you this sort of way to triangulate what might be going on here with their difference in temperature preferences compared to the non-hibernators.
So squirrels and hamsters like the cold, and they'll only go to a hot plate if you raise the temperature on your test plate. And they say this behavior is the same in mice where they've knocked out the gene for TRPM8.
Yeah. So now
Right.
a really interesting molecular connection,
Yes.
perhaps,
Yes.
to this behavioral phenotype, which then allows this foothold to then go deep and look at the evolution of this specific gene to see if there are any clues.
So mice don't like cool. And if you take away this temperature sensor, then they're okay with cool.
Mm-hmm.
So that's clearly implicating TRPM8 and cold tolerance in some way.
In some way. But then imagine if you just delete in real life that gene, there might be a trade-off.
There must be a trade-off.
Of course, and that is that at the same time you do want to sense and transduce a signal involving the temperature that you're in, because there are some obviously adaptive scenarios there involving survival.
So their theory is that cold tolerance of squirrels and hamsters may be caused by fewer neurons with TRPM8 or diminished cold sensitivity of these neurons. And the first thing they did was to use in situ hybridization to ask, well, are there differences in the numbers of neurons that produce TRPM8 in mice versus hamsters and lined ground squirrels?
And it looks like the answer is a resounding no.
No, it's all pretty much the same, right? You cannot explain this difference by a decrease in the number of cold sensing cells.
So then on to the next hypothesis, which is that there's something about the nature of these neurons that is different, or a molecular difference.
That's right.
Mm-hmm.
So they have another assay. They have ratiometric calcium imaging of dissociated dorsal root ganglion neurons. So maybe we should tell people what the dorsal root ganglion is, right?
No, yeah, good idea.
So the dorsal root, I know this only because of herpes viruses, because
Oh, yeah, right.
that is where herpes simplex viruses becomes latent.
Mm-hmm.
And this is a cluster of nerve bodies. A dorsal root ganglia is simply a cluster of nerve bodies outside of the central nervous system,
Mm-hmm.
and they're all along the spinal cord, and they're also at different places in the body. So the DRGs on your face, on the side of your face, is where the nerves from your lips go,
Mm-hmm.
and you get herpes infection on your lip, and it goes up, becomes latent in your dorsal root ganglia, and then when it comes reactivated, it goes back down. You get a fever sore, something like that.
Retrograde and then anterograde trafficking of the virus along the neurons. Yep.
So these are easy to dissect out in animals. You can dissect them out and put them in culture. A lot easier than the central nervous system neurons. But of course, it's these peripheral ones that have the receptors. So
Right.
All right. Then they have a agonist of TRPM8. So an agonist would be a compound that stimulates it, right? And again, TRPM8 is this cold receptor. It's called isolin. Kind of a neat name, right?
Yeah, agree.
Ice.
You know what you're talking about when
That's
the name
right.
of this. It's a good one. Yeah.
So all wild-type mouse neurons are sensitive to isolin, and they're also sensitive to cold. So they have this calcium imaging can measure this response to cold or to isolin.
Yep.
They also can see good isolin responses in some squirrel neurons, about 3%, which show that there's functional TRPM8 there.
Mm-hmm.
And they say even though squirrel and mouse cells had identical isolin responses, the amplitude of cold-evoked response was much lower in the squirrel compared to the mouse.
Correct. And so then we have these great images. So both looking at this sort of readout of the response through direct cell imaging, but then using physiology or electrophysiology to measure the responses.
Right, exactly.
And so that's where we've made that sort of one of those TWiEVO connections that we love,
right?
Is taking sort of that evolutionary idea, but then testing or looking for evidence at the molecular level, in this case, using the neurophysiology.
So whatever this TRPM is doing beyond, sensing the cold, in the squirrel where they hibernate, the response is lower. Mm-hmm. So at a temperature or using isolin, where it activates the neurons in mice, doesn't do it in the squirrels. So they have reduced temperature. They have cold tolerance because these sensors are responding less or at lower levels to cold, right?
That's the idea. So certainly the evidence at this point in the telling of the research project are consistent with that idea.
Right.
But then you can go another step, right, and actually start to try to zoom in on if that's true. What is the molecular basis?
And of course, if you're looking at TRPM8, the first thing you look at is that. Is there something in that receptor or that channel that is making these squirrels less responsive to cold? And that's the obvious thing to do. That's what you have as a handle on this. Mm-hmm. So they cloned the gene encoding this channel from squirrel and hamster dorsal root ganglia, and they can do analyses in culture. They can express the gene in frog oocytes, right? And then they can measure by what we call a voltage clamp. You can actually measure how this channel is conducting electricity across a membrane.
Yeah.
And frog eggs, oocytes, are big, so they're great for doing this, right? You can get lots of them from frogs. You can inject DNA encoding a channel. The channel will be put in there, and then you can easily measure voltage differentials caused by these channels opening and closing, So, and remember, this TPRM8 allows positive charge to go through, so you can measure that with a voltmeter.
Yeah, which is so cool, right? Because you have this direct quantitative readout for how these things are behaving.
And this is something that people do all the time in electrophysiology. There are all kinds of channels that let currents go across. They can be measured, and you can see the effect of drugs or modifications and so forth. And here, it's just the effect of cold on voltage transmittal, of course, or this isolin as well.
Absolute. So then getting back to the not absolute zero, but absolute. So getting back to the evolutionary comparisons. Mm-hmm. So with the idea that it could be something about this receptor TRPM8,
right,
you can then compare it between species, the nucleotide sequence encoding the protein or the receptor itself as it's functioning, and ask, are there any differences that could be consequential when it comes to the shared hibernating species compared to ones that don't? And so here we're comparing rat, squirrel, and hamster, and there are clear molecular differences.
Yep.
Just by doing that comparison.
Absolutely. So here, in the voltage studies, it's clear that squirrel and hamster channels have a diminished cold sensitivity, which is sort of in line with what we saw with the plate test before. And then, as you say, they say, well, all right, what part of the molecule is doing this? Because they are different. The rat, here you said they used, even though the squirrel and hamster are highly homologous to the rat, they have a number of amino acid substitutions in the various parts of the molecule. So they made chimeric channels.
Yeah, I love this approach. And here, I mean, what you really want to do is to distinguish just sort of evolution as usual or sort of neutral evolution, where just by given sort of enough time in enough generations, the error rates of polymerases, other mutational mechanisms, we're constantly insulted or challenged with molecular changes. Many of these or probably the vast majority, are either neutral, they don't have any functional consequence at all, or even slightly deleterious, right? This is maybe why mutation sort of has this almost negative connotation, even though it can be completely the opposite scenario. But when you just do these comparisons and see differences, what you'd really love to do is distinguish between the ones that have perhaps no consequence on function from the ones that do along these lines.
And these are 90% amino acid identity.
Yeah, and that's pretty high, but it's when you're thinking about hundreds of amino acids in a receptor, this one is a transmembrane receptor running through. Yeah, that's a lot of changes, actually. And so using this strategy, this chimeric strategy, where you're basically mixing and matching some of those differences between one species and another, can allow you to rapidly narrow the playing field for what are those consequential changes.
So there are 6. This channel passes, the protein passes through the lipid bilayer 6 times. 1, 2, 3, 4, 5, 6. So they can look at the parts of the protein in the membrane, or they can look at the loops that attach the transmembrane parts as well. Yep. And what they find initially is that if you take the core transmembrane and you substitute that of squirrel with rat, it confers cold sensitivity. So the rat is like the mouse in terms of cold sensitivity.
Right, non-hibernator. Yep.
And then transposition of rat into hamster. Transposition of rat increased Coldevo amplitude sensitivity of hamster, a broad range of temperatures. And so the core is really important, although they have other work showing that the N and C termini can play a modulatory role because they exchange some of those as well.
Mm-hmm. Mm-hmm. Yeah, and actually just to kind of step back to that point you just made, Vincent, about this thing running in and out of the membrane 6 times. And so for receptors, one of the real challenges from a evolutionary standpoint is that those segments that sit, those transmembrane segments that sit in the bilayer are generally, so in order to do that, you need hydrophobic amino acid residues, these sort of lipid-loving, right? Yeah. And so that ends up being, at least the sort of traditional view, is that you can solve that problem of associating or hanging out, inserting these amino acids into a membrane many different ways, as long as those residues are hydrophobic. And so that sort of relaxes the selection so that you can tolerate or get by with many different changes there. And so that is a ongoing challenge, because you look then at these proteins that devoted a lot of amino acids to going through membranes, and immediately you see all of this variation when you do these comparisons. But then to distinguish changes, again, that are just either neutral or don't really matter, that just means you can solve that problem a lot of ways, from the ones that have this phenotype or the difference between that is really sort of a powerful, to be able to do that, to have the functional test is a sort of a powerful way to dissect that.
So what we're talking about here, just to review, this channel responds to cold, and the threshold for response is lower in hamsters and squirrels than it is in rats and mice. So rats and mice, it'll activate, the channel will open and pass ions in at higher temperatures than in the hibernating mammals, and they want to figure out why. And so they find the transmembrane domains, these 6 domains, there are only 15 amino acid differences. Yep. That's great. You always love when there are few. It's less work. Less work. That's right. More clear. So they exchange blocks. They divide it into 3 blocks, and they exchange, and they find transposition of any 2 of the 3 blocks is enough to swap the cold sensitivity of this receptor. So, and that's good. And now they can go down to individual amino acids. And they found 6 residues in the squirrel core that, when replaced by the same ones from the rat, confer cold sensitivity. Yeah,
I really love these studies where you can get down to that sort of single amino acid resolution, where you go from these divergences, which can be millions of years, but then a single change over that time period. We're sort of scaling both time and space in really exciting ways with these kinds of approaches.
And they can, there's 6 amino acids that can swap the property of cold sensitivity. And if they mutate any one of those 6, they can either abolish it or they can abolish the cold sensitivity. So they can go both ways. They can make a channel cold sensitive or resistant to cold, really.
Yeah, exactly. And you can imagine scenarios where selection might act in both directions depending on the lifestyle of the species in question. And so cases where if you had diminished cold sensitivity but aren't, for example, a hibernator, where you could get in some trouble. And then vice versa, if you're a hibernator but then are constantly panicking coming out of that state because it's sort of provoking a response, where that would, because of the evolutionary advantage conferred by hibernation as a life strategy. So really interesting connection.
So in terms of evolution, we don't know who was there first, whether a cold resistant or cold sensitive animal. I mean, it's easier to think of there first being animals in warm places on Earth, like humans emerging in warm places, right? And they weren't so cold tolerant. But then as they moved into colder areas, the animals were selected who could deal with it, right?
Yeah, it's a good point. And, to be upfront, this isn't my field, and so I don't know some of the details. But at the same time, I agree. So you can imagine cases where you sort of get the whole circuit or the behaviors in place. And then as the environment changes over sort of long stretches of time, or then you sort of, fine tune, in a sense, these responses along the lines of what the authors present in this paper.
And isn't it the case that the Neanderthals were actually better able to survive in the cold climates of Europe? And so they moved there, and maybe that, they had just randomly a mutation in whatever their cold sensitivity gene is, and that allowed them to do that, and then that propagated, right?
Yeah, it's an interesting point, an interesting question, and this is like a really exciting ongoing area in molecular evolution, is as we get better and better at getting genomes from these archaic or ancient individuals, to then, as you take a model system or several model systems like this and sort of see if any of the same, since this is independent evolution of different substitutions in that receptor between the squirrel and the hamster, whether or not there are some echoes in our own evolutionary past along these lines. Yeah. I also think, related to that, you can think about the Neanderthals, as you're pointing out, Vincent, or thinking about some of the indigenous populations of our own contemporary indigenous populations of our own species, and so who have long-running histories living in north of the Arctic Circle, for example.
For sure. Yeah. So this channel, TRPM8, it's a major cold sensing channel, and they've shown here that it can specific changes within it can regulate the response to cold. But they say that mice lacking a gene encoding this channel, they have a reduced cold response, but it's not completely eliminated. So there must be other channels that are involved in cold sensing as well. So it's never simple, right?
Of course. Yeah, it's never the whole story. And as you take sort of, a reductionist approach, which is to try to get to this map to this resolution of single amino acid changes, you always have to kind of be careful or remember that, just as you're saying, there's sort of a bigger picture here. And of course, when we think about the multiple inputs, not just the cold through one channel, but, all of the other things that you might associate with hibernation, things like the, shorter amounts of time in sunlight as the days change, and sort of integrating all of these external cues into a molecular response. Yeah, you can imagine things get very complicated very quickly.
And so they think that, if you try and solve it all at once, you're never going to get anywhere. You have to do reductionist, right?
Yeah, I'm a big fan of sort of jumping between both the reductionists. Yeah. I would say a lot of the work in my group and many other labs that think about evolution, so a reductionist approach can be really powerful for moving forward. But you're always you're always sort of making compromises in terms of,
for sure. And you can't say you've solved the problem, right, with a reductionist approach. And many people, unfortunately, do. Oh, solves this issue. But and then something always happens that says it's more complicated than that. So this may partially explain how these animals can deal with hibernation. They have a lower threshold for activation of these TPRM8 receptors in response to cold. Now, my questions are, what is that response, right? We know that ions go in the cell, but what's next? What happens? What is the activation of this channel in a neuron by cold doing downstream, right? And I don't know what that might be. They don't actually talk about that very much. And that's pretty complicated, I think.
Yeah, I think we mentioned this up a little bit up front, but which is that, there are these molecular handles at the sort of input level, which are probably the low-hanging fruit in a sense compared to things getting more complicated. And then they mention at the end here that, in addition to cold sensitivity, they also, reductions in cold sensitivity among the ground squirrels, they've also looked at noxious heat, the other side of the coin here.
Yeah, right.
And also seen diminished heat sensitivity by looking at the variation in another channel, the TRPV1 channel in the peripheral nociceptors. And so that just at least gives you, I think, a hint of that complexity that you're talking about.
Yeah.
There's a lot of moving parts here that are coming together in different ways. And then you can imagine if you select on a change that sort of sets your or alters or modifies the set point for responses, then this could have knock-on effects, right?
Sure,
when you come up against other external stimuli like excessive heat.
Do you think that the response to cold, so let's say you're a mouse and you have these receptors are activated at pretty high temperatures. Do you think the response is just seek out a warmer temperature, or might there be metabolic responses as well to make sure that your core temperature stays the same?
Yeah, I think you could imagine it might be all of the above.
Yeah, probably.
Right? And that, depending on the sort of evolutionary history or the stability of the environmental changes that a species will see.
Yeah.
And and it's a great point that you're raising. And to kind of contrast that maybe for a minute with some of the, the sort of rapid evolution that we associate with our collisions with microbes. So, of course, given our own interest and background, we talk a lot about these genetic conflicts between microbes and their hosts. And to just take a moment to contrast sort of the evolutionary courses that will unfold because of these differences in sort of biological challenges, in a sense. So when it comes to the microbes, it's always sort of a changing menu of variation, right? Because you have these viruses, for example, that might figure out how to replicate in your body, but then then we get into these sort of back and forth or seesawing arms races, in a sense, where you're sort of continue evolving.
It's the same idea, though.
It is.
Cold and infection, because in both cases, it's stuff that's bad for you, and you have to be able to sense it and do something.
So I agree with you there.
Right?
I agree with you there. But let me highlight one difference between a virus and the cold, for example, which is this, is that virus, which itself is a genetic entity that is sampling mutations and adapting.
Oh, yeah. That's got an advantage, right?
Well, or at the very least, it's a moving target.
Yeah, whereas heat is not a moving target.
Exactly.
It's either hot,
Yeah, exactly. And so from an evolutionary standpoint, that has a very different consequence where, we can talk about changing environments, obviously the polar vortex descending on the East Coast even in the last week. But by and large, given sort of evolutionary time, it's a much more stable situation. And so this allows evolution, or we see examples of how evolution has acted in sort of a more almost orderly way. It's still random mutations, but then you do this sort of fine-tuning evolution, which I think is really interesting and one of the things that makes work like this really exciting.
Well, of course, the virus, as you say, and the host can both change genetically, but when it comes to temperature, the temperature changes in response to other things. Although you could argue that human activities are now influencing it, right?
Oh, no, there's no doubt about it. And in fact, we're already seeing really drastic impacts of that. So you don't even have to look at the genetics. You can just look at migratory patterns, for example, birds and other species. Things are changing pretty rapidly. And you can imagine, of course, there's going to be knock-on genetic effects where the individuals and populations that can survive, in some cases, these really radical changes to what has been an otherwise stable environment for millions of years.
Other things, you mentioned heat. So these are all, I would lump them all together, making sure that bad things don't hurt you, right? Infections, cold, heat, pain receptors, the same thing, right? If something is sticking in your arm, you can pull it away because you don't want to leave it there.
Mm-hmm.
What's the other one? The pepper receptors.
Oh,
right?
yeah, capsaicin.
Capsaicin, where, can be bad for you. So you sense it and go, oh, I'm not going to eat any more of that stuff, right? And so it would be really interesting to see, if we could identify the receptors in multiple species, how they change in response to environmental conditions. And would be, in humans alone, it would be really interesting, of course, over time, but in other animals as well. I mean, identifying the players is really cool because then you can do the real stuff where you look at how things change over time.
Totally agree. And I also think that highlights sort of the power of having a good functional assay, whether that's sort of, a difference in electrophysiology or even all the way to behavior, which can be more cloudy. But the reason being, from an evolutionary standpoint, just like you're saying, Vincent, these are potentially pivotal responses in the ability to have offspring. And yet, if we look at the biology, sort of, as we mentioned before, hinted at, the idea that these, for example, the transmembrane receptors that are going to receive some of these insults and sort of transduce them, because there's this sort of relaxed selection in the sense that some of these amino acids and the proteins just need to go through a membrane versus the ones that are, and so to be able to start to discern the difference between the two, you really are stuck, or it's a tough place without having these functional tests.
So here's a more detailed question for you. How do you think these channels, do you think that the channel is the primary sensor of temperature? So at a low temperature, the channel is flipping in some way structurally, and that lets the ions in, and that starts the whole thing? Or do you think there's something above the channel?
Good question. Maybe a little above my pay grade.
I don't know. I mean, it could be either one, right?
Agree. Yeah. Or maybe both in some cases, depending on where the neurons that are expressing these receptors, where they're expressed in the organism, and then also sort of, as that physiology connects all the way through. Yeah,
I mean, because some of the dorsal root ganglia can be close to the surface of the skin, or they can be deep, right?
Agree.
And obviously for temperature, it's good to be closer to the surface so you don't wait too long before the temperature drops and you can sense it. And so I think the simplest explanation is that the channel changes slightly in response to temperature, but there could be other molecules involved, right? That change themselves. It can be very complicated, but as you say, you start at one thing at a time. And then there's the downstream. What happens when the ions come in the cell in response to temperature? What is the outcome? Is it just running away, or is it something more complicated? I mean, seeking a warmer place is pretty complicated, I think, right?
I agree.
I mean, and think of it in a hamster. So they're uncomfortable, and the receptor activation in response to cold is making them uncomfortable. Maybe it's pain or something. So do they just move somewhere else and hope that it's warmer? I mean, in the case of the plate assay, that's what they do. But what do they do in nature? How far do they go,
Yeah, and they're exact. And you have to start to integrate things like memories, or even, these learning with from the mother to the offspring, where some of the cues, or longer term, you can't just, if you move to a warm place, but then it gets, as you peel back the layers of the onion, it only gets more interesting and complicated.
Fascinating, though. Totally fascinating.
Yeah,
and again, this is this behavioral stuff people have studied for years, but only now can we get to the molecular level and understand there's this channel that's involved.
Yeah. I think stay tuned for
more.
Stay
tuned, for sure. Yeah. So they say one thing here that I wanted to get your thoughts on.
Yeah.
They say they want to make a transgenic animals expressing a cold-sensitive ortholog of TRPM8 from the native locus. And they say this should be possible in a few years' time. Mm-hmm. And for mice, you could do that today.
Yeah.
But I guess it's because the squirrels and the hamsters, they don't yet have transgenic technology.
Yeah, that's right. And I think that actually raises a really good point that we sometimes see bubbling up consistently with TWiEVO in the different work that we look at in these non-model species. Mm-hmm. Is that, so with the advent of things like genetic editing using CRISPR and all the excitement around that. So I'm thinking now, we talked about the ants, in a few episodes, some local work here in NYC, and the painstaking work it's easy to say the word CRISPR. Turns out it's much more difficult to do genetic engineering or modifications in some of these wild species. And so even though you might look at a mouse and a ground squirrel or a hamster and say, well, these look about the same, it turns out that investment that we as scientists have made over generations in a handful of model systems is, I think, sometimes underestimated how important that is to have all of those tools and to have refined these protocols and to be able to move forward. And so this, while I'm not trying to throw cold water, if you will, on the idea that there are these possibilities to now move into wild species, make genetic changes and test them in interesting and new ways, to temper some of that enthusiasm with the real work that it takes in order to do that.
Yeah. Now, this paper is largely functional, but there's likely some evolutionary work going on elsewhere that would be interesting
to
look at as well.
Yep.
So this was published in Cell Reports, which is probably a journal you look at often, right? And because there's a lot of microbiology in this journal. And the first author is Vanessa Matos Cruz, Eve Schneider, Marco Mastrotto, Dana Merriman, Svatsolov Bagriantsev, and Elena Gracheva, as you said. And you just saw this and thought it was cool. Huh? You looked at the title and said, "Oh, spicy." I
did, and I love the polar vortex connection. So I'm hoping your cold receptors, Vincent, somehow have a mutation or two that'll allow you to survive these challenges from time to time through the winter.
I don't like cold. I'm telling you, I love the heat.
Interesting. We should genotype ourselves after.
But it's been an age-dependent thing. When I was young, I didn't mind the cold at all.
Yeah.
But I'll live in the Northeast. Okay, I live here. I don't think winter is the most fun season. I don't ski anymore. I used to. Right? But I love the summer when it's hot and humid. I just love sitting outside and, sweating with a cold drink. It's just, and most people want to be inside AC. So I don't know if it's my TRPME.
That's right.
This is a mammalian cold sensor, so presumably it's in us as well.
Indeed. That's right.
Polymorphisms. Now that they know the 6 amino acids that are important, it would be interesting to look at the polymorphisms in human populations, as you said, see if they have anything to do with where you live.
Yep, there you go. And with the grain of salt that some of that variation, is it functional? Is it non-functional? But to imagine,
of course.
Yeah.
Well, you could clone out. You don't have to clone anything anymore. You could take the changes that you see and put them in some tests in vitro like they do here and see if they make a difference. Right?
That's right. Yep.
That's pretty cool.
Yeah. I'm the opposite of you, actually, Vincent. So I prefer...
You like cold?
Well, I wouldn't necessarily love, I don't want to freeze, but if I had to pick between being too hot or too cold, let's say when I'm trying to not hibernate but trying to sleep, I feel like if it's cold in my environment, at least I can put more blankets on. Or if it's hot, of course, you can turn on the AC or the fan, but it's a slightly different.
Well, I think sleeping is easier in a cooler temperature for sure. Okay, I like to have it at 66 at night, and I always have conflicts with my wife because she wants it warmer. But that's because you're from Minnesota, and your ancestors came from Scandinavia, presumably, and they have some mutations that make them like the cold.
There you go. Someday we might know the answer to that question. Yep.
Anyway, that's a cool paper.
Yeah.
Anything else you want to say about it?
I think that covers it.
Yeah, that was pretty cool.
Yeah.
Let's see. What do we have next here? We have some email.
Let's move to the mailbag.
Why don't you take that first one?
Yeah, sounds good. Stephen is getting in touch with us, and thank you, Stephen. We've been going back and forth, and I'm glad you took the time to write to us. So Stephen says, Vincent and Nels, I really enjoyed the most recent TWiEVO on the importance of DHX9 in defense against endogenous ALU RNAs, and particularly the appearance of ADAR, given recent interest in our lab. It's been known for some time that genetic ablation of ADAR is lethal to mice at the embryonic stage, but that this could be overcome through ablation of MDA5 or MAVs, two other proteins, though these mice still aren't exactly healthy. Recently, we discovered we couldn't even knock out ADAR in human A549 cells. We had previously found that RNA-cell activation is the primary mechanism of cell death after introduction of poly-IC. It's a sort of nucleic acid mimic, and decided to test if RNA-cell ablation would allow us to knock out ADAR. Unlike most experiments, it worked and was published earlier this year in eLife.
So that means you have to have ADAR to respond to RNA.
Correct.
Because if you knock it out, then the RNA kills you.
Yep.
Right?
Yep.
Through these RNA sensors, right?
That's right. So the sensing, in many cases, foreign nucleic acids from microbial invaders. We did a few other things to really nail down that RNase-L was responsible for the lethality observed in the absence of ADAR. Specifically, we made a cell line expressing the NS2 protein of mouse hepatitis virus, a well-characterized antagonist or blocker of RNase-L. In this cell line, but not a cell line expressing a mutated inactive NS2, has also successfully knocked out ADAR. Additionally, we determined that cells lacking ADAR produce more 2,5-A, a small molecule required for RNase-L to activate. I'll avoid going on any longer than that, but thanks for highlighting the really cool and growing field around modification and sensing of endogenous RNA and overlap with antiviral responses. Thanks as well for making TWiEVO happen. It's been a pleasure meeting both of you over the last several months. Best, Stephen.
Stephen's at Penn, right?
Correct. Yep.
Where did you meet him? There?
I met him there. I also met him at, speaking of cold sensitivity, at a meeting near Taos, New Mexico. And we did some skiing together, actually, one afternoon. That was really fun.
This is a good story. And, hey, maybe RNA cell ablation, but, it's not trivial to do it. So it's cool that it worked.
Totally agree. Yeah. And keep an eye on Stephen and the crew at Penn who are continuing to make these really exciting discoveries along these exact lines. Yep. Thanks, Stephen. Great to hear from you.
All right. The next one's from Anthony. The question of the advantage of size came up on TWiEVO 26. So that's our episode with Vinny about elephants and whales. Why don't they get more cancers, right?
Yeah, right.
But why are they big to begin with? Here are some impressions, speculations. 1, the ability to support a bigger brain. That appears to be the reason for the increase in size in hominids. 2, due to the vastly reduced surface area proportionally, gigantic animals will cool much more slowly, clearly an advantage for the whales that inhabit very cold water. Might this have been an advantage for the dinosaurs too? 3, taller animals can graze on trees and see food water predators that are far away. Might the large size and blubber of the sperm whale enable the deep water dives necessary to hunt the giant squid? There was mention of reduced predation. This could quickly result in an arms race that produces all giant animals, predators and prey alike. That's an interesting points. Yeah. And Anthony wrote to us and Vinny Lynch, so Vinny answered, Hi, Anthony. I think there are probably lots of reasons being big is both good and bad. Whether a species evolves to gigantic sizes probably has to do with how those advantages and disadvantages are balanced in that particular lineage. Being big is great, but it takes a long time to grow big, time in which reproduction isn't happening. So if you die before you reach maturity, there's not much help there. That's a good point.
Yeah, that's right.
I'd bet there's probably an upper limit to predator size. All the really big things are or have been herbivores. My guess is that it'd be hard for super gigantic predators to get regular access to food or something similar, but being big is definitely a good way to stay warm.
There we go. Sort of an echo from today's episode as well, right? With this, and actually, I think a nice reminder in a sense that, we have been focusing on sort of some molecular changes of a receptor, but also these other sort of physiologic or even body plan outcomes that might have impinge on your ability to live in extreme environments.
So, if we could identify selection at the level of the TPRM8 gene, that should be removed in a really big animal where they're much, much less influenced by cold, right?
Yeah, good idea. Although then, assuming, I guess, that body size has been stable over some evolutionary course, and so that could be a moving target in a sense that, at least from a millions of years point of view, that things could change. And so just making sure that you're trying to account for all of the complexity can sometimes be pretty daunting. But
the most successful organisms on Earth are tiny. Viruses, bacteria, insects, right?
That's right. How are we defining successful in numbers?
Just numbers.
Yeah. No, agree. Or you could define it maybe by the weight of genetic material. And I think, that gets at this idea, the imperative of replicating or, the gene persisting. And so, but under both counts, I think you're right. I agree with you that it's the small stuff that's really done well. Okay. You're next with Anthony again. So Anthony writes on James Joyce's powder pigeons simile in Ulysses. Joyce clearly demonstrates that he was familiar with the appearance of pouter pigeons. I wonder, how did that come to be? When young, did he or an associate keep pigeons? Did Joyce browse the live animal markets? I also wondered why he used an image that few would likely recognize. A Joycean gave me a likely answer to both questions. In Joyce's era, every educated person would have studied Darwin and would be familiar with the experiments in atavistic reversion in pigeon crosses. Another nice point from Anthony here. Yeah, it's interesting. So the pigeons, and going back to, I'm blanking on the TWiEVO episode, but with my great colleague, Mike Shapiro, who's doing a lot of evo-devo, evolutionary developmental approaches with the pigeons, where there's been these great and ongoing hundreds of years experiment going with pigeon breeders all the way back to Darwin's time. And so I think that does connect it through the fact that Darwin himself became fascinated with pigeons and the different morphological forms that will come out of these crosses as people sort of are keeping and collecting and doing artificial selection for the things that kind of caught their eye. That will, it's interesting how these things sort of echo through not only the science, but, society at large, right?
Yeah, for sure.
Yeah.
Anthony provides a little quote from Ulysses. He provides a link to the full text of Ulysses, which you can get at the Project Gutenberg. And there's this little exchange. Bloom, yes, ma'am. Marion, ti trema un poco il cuore. Is your heart trembling a little? In disdain, she saunters away, plump as a pampered pouter pigeon, humming the duet from Don Giovanni. It's pretty funny.
That's great.
Powdered. So this is from Mike, what was his name? Shapiro's episode, long time ago.
Long time ago, one of our first. So he has an interesting, you can go back and listen to the episode. I forget, it's probably in the first 10, collection of first 10. He has a really interesting history himself, going from more paleontological background almost, to then spending a lot of time thinking about the stickleback fish, which is another really great system. Well, we haven't already, but I would suspect that we'll get around to some stickleback stories, which have a great place in evolutionary biology. But then when Mike started his lab at the University of Utah in the biology department, he made a big left turn to this pigeon project, which has really taken over, and they continue to just pour out really exciting work. I think he just had a paper published even a couple of weeks ago, so definitely worth checking out.
Was that the only pigeon TWiEVO we have? Somehow I feel that we had another one. I don't know why.
Well, we've been covering a lot of...
Pigeons Show the Way.
Right, yeah.
That was TWiEVO 25.
Yep.
Remember the passenger pigeon genome.
Yep. Another Shapiro, not related, but Beth Shapiro's lab at Santa Cruz, right? Yep.
Yeah. All right, take that last one, Nels.
Okay, this is from Irene. And Irene writes, Dear Nels and Vincent, it is currently 43 Fahrenheit, 6 Celsius in Durham, North Carolina. But since I'm almost always in the greenhouse, it's perpetually in the 70s Fahrenheit, 20s Celsius. Working in the greenhouse has meant a lot of time to listen to podcasts, including TWiEVO. I have a few comments, suggestions, questions, which I will list below. Let's take these in turn. Number 1, I particularly loved the episodes with Corey and Josh, who highlight the importance of natural history museums. Thank you for bringing them to light, especially since a lot of those museums are closing down. So yeah, I agree. Let's take a break or a moment to just address this. So actually, Josh was here in Vivo with us some months ago. Really fun interview. And then Corey, who's at the Field Museum in Chicago. Glad that you like these, Irene. I agree with you on all fronts here. Number 2, I also like how several of the guests have emphasized the importance of having a question or questions to drive their research. Even though sequencing and functional genetics is becoming more accessible, it still takes lots of time, money, and other resources to develop tools to do some functional work. It is also sometimes hard to do it with certain species, and it is worth asking whether doing a functional experiment, like knocking out a gene using CRISPR, will tell us anything new or important. 100% agree, Irene. In fact, if we rewind a few minutes to our conversation with today's work, I think we're on the same page exactly. Number 3, coming from the perspective of a budding plant evolutionary biologist, my question is, where are the plants? You've covered research from a myriad of species, and while there have been some mention of plants, I keep wondering when plants can be the focus of the conversation. Perhaps this is in the works. If you are interested, I would be happy to suggest a few papers and people who would be fantastic guests.
I think we should take her suggestion, right?
100% yes. There is incredible evolutionary biology and function, connecting it through to function theme here in plants. And I agree, we're a little slow to the game, and I'm somewhat embarrassed that we haven't already covered the plants, but we'll remedy that
So
situation.
Irene, give us some suggestions.
Yeah, please. We'd love to hear.
Yep.
Number 4, however, if plants aren't really in the pipeline for the future, may I recommend 2 plant-related podcasts as my listener picks? Of course. So number 1, Irene highlights a podcast called In Defense of Plants. It's hosted by Matt Candelas, a PhD candidate, and his goal is to cure plant blindness. Side note, I have no clue how he is doing this. In working on his PhD, I can barely find time to do my own research. There is a blog, a podcast, and a video series. I haven't looked through everything yet, but everything looks fantastic. I have listened to a few of the recent podcasts in which he talks to scientists, landscape architects, horticulturists, etc. I think this is one similar to the format of TWiEVO and is worth a listen. In Defense of Plants, interesting. And we'll of course put up the link to that. The second one that Irene lifts up is a podcast called The Taproot. It tells the story behind a plant-related paper, the collaborators, the challenges, the compromises, etc. It feels like a shorter version of TWiEVO, focusing not necessarily on the science in the paper, but the process of making the discoveries and getting work published. The first episode is quite fascinating. I'll check out both, Irene, and we'll get those links up to our listeners as well. So thank you a bunch. And then the last point from Irene's great note is number 5. Finally, having heard some panel discussions on how some podcast taping occurs at meetings, I was wondering, would it be possible to have a TWiEVO taping at a future evolution conference, perhaps in 2019? I only say 2019 because the 2018 meeting will be a joint meeting that tends to be very structured. Or perhaps there could be a recap of talks you attended, kind of like All Songs Considered, recapping South by Southwest. Mm-hmm. It is also likely that you do recaps already in other podcasts, but I thought I should share the idea anyway. Thanks for all you do. I look forward to future episodes. Best, Irene.
Do you know this meeting?
I do. And in fact, so I will be at the Evolution Conference in August 2018 in Montpellier, France. Montpellier, yeah. And so I agree, and we do. You have a long history and growing history of doing panel podcasts.
Oh yeah, at different meetings,
exactly. Yeah. And so I would love for us to start to expand our playing field in these ways. And so,
well, if Irene knows the organizers, or if you do, float the idea of doing it in 2019. Where would it be? Do you have any? Are they always overseas or?
No, it moves around. So there was one actually not too long ago in my backyard at Snowbird in Utah. It was in Portland, I think, last year, and then it will be in...
I think you had asked if I could go to the Portland one, wasn't it? Or was that the Seattle meeting? A different meeting in Seattle?
Yeah, different meeting. Okay. But we'll continue, obviously, to keep our eyes open for opportunities like this. And so stay tuned. Actually, that opens up... I want to plug a meeting that I'll be hosting with some colleagues at Utah. Save the date out there, everyone, for May 9th through 12th. This will be a meeting sponsored by SMBE. This is the Society for Molecular Biology and Evolution. They have every year now, they have a growing slate of what they call satellite meetings. These are smaller meetings. So the annual meeting is more than 1,000 scientists now, where this is about 1/10 the scale. Mm-hmm. And we'll be covering the topic of molecular evolution in the cell. So people who are mixing cell biology with evolutionary biology, somewhat along the lines that we hear about in TWiEVO. And I'll get a link to the meeting website. It's still coming together, but we have a great lineup of speakers, including some like Nicole King, for example, who you've heard in TWiEVO over time. So take a look at this as this comes together as well.
Neat. Yeah, we'd love to do that, Irene. So you can hook us up or suggest it or start the idea rolling.
Yeah.
And thanks for your listener picks.
That's right. We'll get those up.
Nels, you have a pick of your own?
Yeah, and actually this is a really nice tie-in, I think, to some of these other podcasts. So I have a link to a blog, but it itself has links to some other podcasts. These are actually undergraduate podcasters who took a course on evolution. This is a class at Stonehill College. I'm embarrassingly blanking on the name of the professor who runs the class, but we'll get that up there. And so if you go to this link, everydayevolution.org, there are a series of short podcasts that are part of this class's projects, or graded on that. And so students in the class have taken research or papers, very much like we do here on TWiEVO, and then put together a short podcast trying to capture almost a kernel of interest for a broader audience. And I really have to say it's inspiring to listen to these because they're so professionally done. It's, on the one hand, inspiring. It also makes you realize, what we're doing, we could be replaced in about 1 minute by many minutes, right? And so even the great introductory music in these as well, and sort of tying it together in a fun way. So some really cool podcasts on things like the evolution of bitter taste reception, sort of echo from today, and all these other things. And so that's my pick of the week.
So this is a evolutionary biology course, Stonehill College, and Dr. Bronwyn Bleakley and Dr. Nick Block. Those two seem to be the professors here.
Yep, that's right.
That's pretty cool. And there are multiple students on each podcast. 1, 2, 3, 4. So they do them all together. Yeah.
Exactly.
That's cool.
Yeah,
Great.
Yeah, really fun. How about you, Vincent? What's your pick of the week?
Well, I have an interesting pick which I got from friend of TWiV, Islam Hussein, who does the Viral Vlog video series, which is a virus series in Arabic over on YouTube.
Yeah, very cool. And
he sent me for my birthday some viruses that he got at Target. And these you can buy at Target. He sent me an excited text message last week and said, "Look what I found at Target. They have viruses." And I found them online as well.
Yeah.
They're called gold faceted tiles, decorative wall sculpture. But they're half icosahedra.
Yeah.
That you're supposed to hang on your wall. And they are, the way some capsids of viruses are built. And so he sent me a pair for my birthday, along with a nice card that his son Adam had drawn of viruses, which is just beautiful.
Oh, wow.
And it's funny, the pick is the link in the catalog because the description is funny. Add some shining style to your space with the gold faceted tiles decorative wall sculpture.
Yeah.
Each piece features a flat side that lays flush against the wall, and the other side is covered with raised multiple facets and a gold finish for optimal shine. Place one on either side of your front door or add to your photo gallery wall to create a unique look. Of course, I should put these on my wall because it will identify me as a virologist.
That's right. Great to see the virus kind of capsid structure showing up here, and for their beauty.
Yeah, I mean, in fact, if you look to your right there, up on the second shelf, see the big icosahedral thing?
Oh yeah.
So that's a wireframe icosahedron I bought at West Elm.
Oh yeah.
I saw it there, and they have all these these companies sell decorative things you put in your house, and it turns out that icosahedra are pleasing.
That's right.
It's a regular sculpture. It's a regular object, right? And so people like it. And it turns out, of course, that viruses are icosahedra as well. I have something new on my desk here. Since the last time you were here, Nels, I learned on a trip to Indiana University how to build icosahedral capsids from buckyballs.
Oh, wow. Look at this.
So I had previously made helical capsids, which is easy, right?
See
you just
that.
wrap them around. But there's a fellow out there that was making them out of buckyballs. And the cool thing is, now the one you have in your hand, that's a T=1.
Yep.
Where all the subunits are pentamers.
You can see
5
balls per
5 balls, right?
surface. Yep.
And you see it's small. But look at this one. This is a T=3. This is made of pentamers and hexamers now.
Wow. Yeah.
And I color-coded it so you can see the difference.
Very cool.
And that to make it even bigger particle, all you do is add more hexamers.
So cool.
And it gets bigger and bigger. So I think this is a beautiful teaching tool.
Great point. Art meets science. Science meets art. It's a great connection.
Love it.
And happy birthday, by the way.
Thank you.
Yeah.
Thank you very much. All right, let's give away a book.
Great idea.
So I have a copy of Viruses, Agents of Evolutionary Invention. It's written by Michael Cordingley. And he sent this to us, as I mentioned earlier, and he wanted us to give them away on TWiV and on TWiEVO. We gave one away on TWiV already, so...
What a good idea.
And the chapters are great. 1, obligate parasites of cells. 2, viruses, genes, ecosystems. Potentiation of bacterial diseases by phages. Viruses and higher organisms. The flu. And it goes on and on. endogenous retroviruses. So you could own this book. We would send it to you.
Fascinating. And author autographed, an inscription.
Autographed. Yeah, a specific TWiEVO, to TWiEVO listener, and he's got a little note and he signs it as well. So if you're interested, you have to send us an email, TWiEVO, TWiEVO, at microbe.tv. And as the subject line of the email, you just write viruses, because that's the name of the book, okay? And we'll see it. And I don't know how many we'll get, but we'll pick one out at random. But, in your email, since you're sending us an email, just tell us a little bit about yourself. What you like about TWiEVO, what you do, anything. Or you don't have to write anything. You could just say, I'm entering the book contest. But we'd love to hear from you, wouldn't we, Nels?
Absolutely. We'll take all comers. And,
all right.
All comments, positive, negative, everything. Sure. And check out this book. It looks really fascinating.
TWiEVO is you can find it at Apple Podcasts, microbe.tv/TWiEVO. If you have a favorite podcatcher, subscribe to it. Do you listen to podcasts, Nels?
I do.
What do you use to listen to?
I think I go through the Apple Podcasts.
Yeah, program, right?
Easy enough. Yep.
Yeah, that's on every iPhone.
Mm-hmm. One of my favorite podcasts is TWiEVO, This Week in Virology.
Really?
I really enjoyed. I guess this is 1 or 2 episodes ago. The giant virus, BSV, Bodo Sultan's virus.
Cool.
And we'll get back to that maybe this afternoon as we're doing back-to-back podcasts.
That's right. That's right. So you can subscribe on these podcatcher apps on your phone or tablet and get every episode. It helps us a lot if you subscribe. And if you're on social media, Twitter, Google+, Facebook, that's where I usually announce the release of our podcast. It would really help us if you liked or retweeted whatever I do, or Nels as well, to kind of amplify the message for our podcast. We'd like to get more and more people listening because we think, as you have told us, that they're useful. They're educational.
That's right.
So please do that.
Thank you.
And if you really like us, please consider supporting us. You can go to microbe.tv/contribute for ways that you can do that. Questions and comments, as well as your book entry, TWiEVO at microbe.tv. Nels Elde is at cellvolution.org. On Twitter, he's L Earlybird.
Hey,
Nels, thank you.
Thank you. Great to be in Vivo.
Yeah, I'm
Always
glad you thought of coming here while you were in New York City.
Yeah, of course.
Many people do visit, and they never say, I'm here, I'd like to do a podcast, so I appreciate it.
Yeah.
And what's your next trip, Nels? Where are you going?
Good question. It's not going to be long before I'll get back to Utah, and then I'm heading over to San Diego.
Nice.
There's going to be I think the microbiology training grant is going to have an all-day symposium, and I'll be part of that. I'm really excited for that. And then I will be hosted by a great virologist, David Wong, at Washington University in St. Louis right after. So on the road for a while.
Good.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. They're at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.